time since Monday, we've heard a lot about regulatory and regulations, and then gene drive. Let me chip in this to at least calm the nerves down a bit. So, with all these discussions, let me just create a scenario of a world where we have mosquitoes that are causing all these devastations, killing so many people, mostly our children, around 430,000 children being killed from Africa every year. And then we have these food security issues with so much money that will be needed to feed the people if we lose the pollinators who would help us get this food. And then most of the global living crops are also dependent on some of these issues. And then we have the mosquito in the middle. So here the regulation comes in. The scientists are here saying because of this, let's get away these mosquitoes and then we are free of the diseases. And then some of us, the ecologists and then the biodiversity people would also be saying that no, this is part of this ecosystem service, providing this ecosystem service. And so if you just get it out, we are going to be hungry. So where do we find the balance? Let's see if we can get a balance for this. So we are asking the question whether suppressing this particular species, anopheles gambiae, that is causing all these devastations, could impact on pollination and then affect the reproductive success of some plants, which would lead to the end that we are going to be hungry because of pollination issues. So where did we start from? Starting, we don't know or we didn't know whether the mosquito is visiting a plant or which particular plants they are visiting. The literature is short of information on this, though it has mentioned some other species, including the killers and then the Aedes as possible pollinators or pollinators of some plant species. There hasn't been any literature on anopheles gambiae as a pollinator or actually visiting plants in the wild. So we set out on a gray level, collecting all plants around our lab and then trying to test them. The assumption was that when they close or when they come out of the pupa stage to adulthood, they might not have much energy to fly long distances looking for food. So they would obviously hook onto one of the plants around. That is if they need that to survive. So we collected all plants in food blooms around the lab. That is the Mosquito Ecology Research Facility by Target Malaria, University of Ghana. And then we tried to see whether the mosquito would visit these plants. And because we didn't know which part of the plant it was going, we just took the branch of the plant that had flowers on them and then released some 40 newly hatched mosquitoes into the cage and watched them overnight to see where they were visiting. And what did we find? We found out that they were visiting mostly the leaves and then the stalks and all other parts, but few of them visited the flowers of specific plants. This is castor oil and then mango plants. So what we found was that they significantly visited the leaves and the node stalks and not the pollen. We decided to find out whether if we gave them only flowers, they would be able to visit, and then they will pick up the pollen, which would lead us to the field to see whether they are actually visiting or not. And what did we find when we did that? So we took flowers of the same plants in cages, released the same mosquitoes into them, and watched them under the microscope the next morning to see whether they were carrying pollen. And our observations was that they were visiting every flower of the plant we gave in. This is a plant that closes the flower abutilon. The flowers were completely closed, but they were still trying to find something, telling us that they actually go to plants to find fluid or whatever they will get for it, and not specifically to go for the flower, which could lead to pollination. So, observing them under the microscope the next morning, this is what we found. So many pollen on them, on the nectar, on the legs, wing, back, everything, indicating that when they go to the flower, they would be able to pick up pollen. And then when you observe them under the mic, you would see this pollen on them. So this led us to the field to find out whether they were actually visiting or not. So that we will not just say that they are visiting the field or they are possible pollinators. We wanted to be able to evaluate how much importance their pollination or their contribution to pollination was. So we set out to do a network analysis to see their role in pollination, and then we sampled from 2 communities in southern Ghana, and this is how we did our sampling. We actually have 4 microhabitats within the communities: farmland, in-village vegetations, grassland, and then forest. We sampled from each one of them all insects that were visiting flowers in the day and then in the night. We specifically gave some time to sampling for anopheles gambiae, looking for their interaction with plants. We spent as much as 608 hours looking for these interactions, but we found 0. So what is this telling us? We went on to see that, okay, if they visit flowers and they pick pollen and it could be detected on them, then let us collect the mosquitoes themselves and observe them under the mine to see whether we could see some of these pollens on them. So we collected mosquitoes from their hideout in rooms, in abandoned houses, and so many places. And we collected as many as around 694 individual anopheles gambiae from outdoors and indoors. And then we observed them under mine to see whether we would find pollen on them. But before I tell you the results of that, let me just share with you what we found on the field when we were looking for mosquito-plant interaction. The closest we could find was this, which we identified as Culex species, that was closer to the petal of this particular plant, Melanthera scandens. And then in our lab experiment, we included this particular plant, and then some of the anopheles gambiae were going to the petal and not on the flower, or yes, on the flower is where it could pick pollen. And then we found some of them on grasses and then on leaves, among others. So, we never found any anopheles gambiae in the field plant interaction, neither did we find any pollen attached to any of the 694 anopheles gambiae we collected outdoors. So we put this to a network to evaluate the overall importance of anopheles gambiae, even if they were involved, and how that could impact on pollination, if so. So this chart here, we have the top row, that is the bars on top being the insects. That's the insects up here. It's okay, so these are the insects, and these are the plants that they would be interacting with. And these bars are actually how many times this particular insect is visiting plants in the whole area. And then the line here kind of depicts the number of times this particular insect visited this particular plant. So the more you visit the plant, the more you're likely to pollinate that plant. And the more plants you visit in the area, the more important you become as a pollinator. Then it will mean that you are actually pollinating so many plants, more or less the general characteristics. So we found out that obviously we didn't see any anopheles gambiae direct plant interaction, neither did we see any pollen on them. So obviously they are out of this, and that tells us that in the pollination community they are actually either a peripheral or they are not part of the whole show at all. So this tells us that anopheles gambiae, if forced onto plants as we did in the lab, they are able to pick so many pollen that is comparable with some insect species that pollinate some plants or of Asteraceae and then others, and is also comparable with Culex ppn. The amount of pollen that Culex ppn picks when it visits these plants that it actually pollinates. That has been shown by Peach and Grice. And then all we want to say is that anopheles gambiae suddenly or may not be visiting flowers at all, or even plants, so to say. this is justified by searching for them for as many as 608 hours. We never found any of such interaction, and I must say that we did a nice survey knowing that they are nocturnal insects, and the most likely time that they will be visiting plants will be in the evenings. And then we also collected 694 and never found pollen on any of them, but we were able to show that in the lab, when they visit plants, they pick pollen, and these pollens are able to be detected on them the next morning. So all we are saying is that they might not be, or they are not important pollinators from within our research area. So now we solve the myth for the regulators and all of us. Please, if you are only thinking about them as pollinators, you can go ahead and then reduce their population so that we save our children from dying. Thank you.